Great. Seems to be my turn now. Yeah, I would like to thank the organizers and especially my former PhD student, Mano Rizayeva, to inviting me here. And I'm really impressed to be here. I have been in Tashkent 40 years ago, and it was kind of cultural shock to come here, but in a very positive sense. It was really impressive how the city has developed, how also even scientific institutions have developed. It's a pleasure to see that. And yeah, and I'm proud to be here the first to give a lecture. And the lecture is about genetic engineering. So genome editing is, let's say, the most modern way of genetic engineering. It is no longer introducing genes from one species to the other. It is just writing in the genome and to improve performance of plants, in our case, plants, to the plant's benefit, and in the end also to our benefit when we consume or when we use the product. So just for your understanding, I'm German. I come from Germany, from the Leibniz Institute of Plant Genetics and Crop Plant Research. I'm head of the group Plant Reproductive Biology. And Vano is still a member of this group, even though she went to the United States meanwhile. Having a new job, she is still guest researcher in my group, and I'm proud of that. So, how do how does genome editing work? Um, what we have to do is to introduce into the cell an enzyme which cuts the DNA of the plant. Um, and the system is a two-component system. It consists of an enzyme, leads the DNA, and an RNA, which guides this enzyme to the site where the DNA has to be cleaved. And as you can see here, the blue structure is this enzyme. It is CRISPR associated, its name. And the guide RNA, again, consists of two parts. The light red part is a specific sequence which fits to the target sequence in the plant genome. It's complementary to the target sequence. And the wine red part is a kind of scaffold structure which interacts with this enzyme. So these two guys form a ribonucleoprotein and act together. And when we have introduced these two parts, typically by introducing genes which encode the enzyme and another one which encodes the guide RNA, once this complex is present in plant cells, it goes to the nucleus and there screens the DNA for the target motif. What first happens is that the enzyme looks for a very small motif, which is actually a double G in this particular case of the Cas9 version. And the enzyme stops there, and then the guide RNA at the opposite strand looks for complementarity in its 20 specific nucleotides. And only in the case the guide RNA finds a match, it tells the enzyme, okay, now you can accommodate here and activate your function. So, and the Cas endonuclease has two cleavage domains. So the DNA, you may all know, consists of two strands, and each strand needs to be cleaved. And for each strand, there is a cleavage domain in the enzyme. And these cleavage domains are activated as soon as a guide RNA is recognized perfect target. So, and after cleavage, we have this double strand break in the DNA. And that's actually all what we do as biotechnologists. The rest is provided by the plant cell itself, because plant cells are used to cope with double strand breaks. Every now and then, those things happen spontaneously, and every organism needs to be capable of repairing DNA. And we can now wait that this cleavage is repaired, And in the majority of repair cases, the former sequence is restored perfectly, but since the restored target is cleaved again,
they need to translate this, so they will into Russian. Please just give a break every
all the time.
yes, but just I think just for lots of more views.
Okay.
Yeah, but human look really short. It's look like it.
Okay.
Yeah.
Yeah. Thank you, whoever has translated. It's very impressive that you keep all that in mind. So to go on, once the DNA is cleaved, we have cellular DNA repair mechanisms which will fix the problem. Typically, the old sequence is restored, so there will be no effect of the break for the genetic information. But as you all know from your family members or craftsmen coming at home when a certain item is repaired 100 times, mistakes happen, and we have the time to wait for that. And this, you see, then that nucleotides can be deleted in this repair process or inserted, or we have certain nucleobases exchange. Okay, and with that, we have already fulfilled the major principle of this technology. Insert mutations, not randomly in the genome, which we all know from spontaneous modifications over evolution and domestication, or chemically induced mutations, or irradiation-induced mutations. So these older forms of mutagenesis appear to happen randomly in the genome, and the progress in our technology is that we can predict where exactly a mutation will happen. So to explain where this biological principle comes from, we have to go back to bacteria. Actually, the system was evolved in bacteria to combat foreign DNA, mostly in the form of bacteriophages, which are viruses. And what you see here is a bacterial cell. And in the cell is attacked on top of a bacteriophage, and this bacteriophage releases its DNA into the cell. So, and the bacterium is capable of cleaving this introduced DNA randomly. So it recognizes any foreign DNA unspecifically and cleaves it into smaller pieces. And these smaller pieces are then taken and introduced to the bacterial genome. And this you can see with this kind of numbered pieces of DNA introduced into the genome. So, and after having introduced a couple of those sequences, you see it looks like a library. So the bacterium can keep in mind specific DNAs of viruses. And this system now is so smart that these pieces can be expressed in the format of RNAs. And these RNAs then will serve as a guide RNAs, as I have described for you for the genome editing system. And as I have explained formerly, this guide RNA interacts with this fast endonuclease. In this example, it is more a complex of different endonucleases, but the researchers have, of course, selected a system which consists only of one enzyme to simplify the enzyme. The principle is the same. We have a complex of guide RNA and fast endonuclease enzyme, and this specifically finds DNA with the same specificity as the former infection. Now, if you have a second infection, this foreign DNA will be recognized much more specifically and can be degraded much faster. So this provides immunity to the bacterium to arrange macrophages. So here are the steps I went through. I will skip that now. Coming to the point of how we implement this technology. And as I told you, we have to introduce typically these components, fast endonuclease and guide RNA as transgenes, at least for a while, as long as they are needed to modify the genome. And for that, we use conventional transformation techniques. And in my lab, we have a focus on monocots. And Barno once broke this convention, and he started to work on a dicot, which he will talk later about her story. And I will confine my talk to monocots, or maybe more specifically to grasses and cereals. So what you see here at the left-hand side is an immature embryo of barley. And this immature embryo can be induced to form a callus. The callus forms then secondarily differentiated structures, and we can regenerate plants in vitro or many plants out of one embryo. A second system we are using is immature pollen. Immature pollen, it's a unicellular stage called microspores, and these microspores can be reprogrammed to undergo cell proliferation, and from these proliferated structures, we can also regenerate plants. That's later. Yeah, let me first explain before you translate it. So the third picture is an isolated ovule with a little bit of pericarp on top. And the ovule is this kind of transparent bubble. And in this ovule, an embryo is developing. And what we can inject by the capillary, which is shown coming from the left, to this ovule, we can inject agrobacterium, and the agrobacterium then transforms cells. It encounters, and amongst others, all the cells of this developing embryo. And when this embryo then further develops into a plant, we can produce a transgenic plant. The last example is just for the sake of completeness. We have the model grass Brachypodium distachium, which is not a cereal, it's just a grass, but very comfortable to work with. And in this, we have a system where we just cut some segments of the shoot base, and this shoot base forms the colors, and then we can regenerate plants. So, and all of these systems somehow develop a regenerable structure. And as a representative, you see here below the colors, forms leafy structures at its surface. And to indicate transgenic cells and tissues, we use a fluorescent protein from a jellyfish. And this fluoresces then in green, as you can see here, and some of these surface structures. And this is an indication that some transgenic plants will develop from this. So under these pictures, I have indicated in which species we are capable of using these methods. And these letters, these capital letters A and B, indicate whether we can do it using agrobacterium or the ballistic method, where we introduce or we shoot gold particles which are coated with DNA into the cells. So, and the last picture shows you a spike which has been formed from a primary transgenic plant. And here you see already what we could interpret as the babies of the next generation. These are the grains. Yeah, and these grains, as you can see, follow a certain segregation. So we have non-fluorescent and fluorescing grains. And of course, the genetics are a little bit different as compared to classical Mendelian segregation, but it's similar. So now we switch to the first example of genome editing, using all these kind of introductory tools or tools I described. And when we started to use this technology, we thought, okay, let's produce some examples almost everyone can understand and can see how powerful this method is. And here we have the example that barley, you may all know barley, has two basic types. And one group has 2 rows of grains along the rachis, and the other one has 6. And as we knew, the gene which is responsible for this difference, which is a homeobox transcription factor, we know what gene we need to knock out so that the lateral spikelets, as seen here at the right-hand side, which are only rudimentary in the 2-rod system, are fully developed in a 6-rod barley. And of course, everyone can conceive the yield potential of the 6-rod barley is much higher than of the 2-rod barley. We just switch by genome editing between these 2 forms. That's fine. So, and this is how we did it. Here you see the 2-rod barley, which is cultivar golden promise. It's one we are working most with. It's a model because it's easily genetically modified. And it has a function of this VRS1 gene, which regulates the development of the lateral spikelets. In the foreground, you can maybe imagine that there are these kind of rudimentary spikelets. So, and what we did is we had a look at this gene and we selected an appropriate target motif where the gene can be mutated. And we transformed this golden promise with the construct. And here you see the target motif within this VRS1 gene. And as you can see, just as an example, in the above line, you have the wild type sequence, which is the functional sequence. And we obtained several mutants, and just one mutant had a deletion of one nucleotide. So this 1C is missing in the mutant, and this results in the abolishment of the whole gene function. So the repressor of the lateral spikelet formation is no longer functional, and the result is a 6-rod barley. This is the tiniest genetic modification you can do. Just delete one nucleotide, and you have the tremendous difference 2-rod to 6-rod. Yes. The second example is to create naked barley. For us, European naked barley is something not everyone is aware of that it exists. For you Asian guys, it may be much more common. Am I right? Naked barley. So I know in some Asian region it's really common food. So barley, actually, we cannot eat humans because the hulls around the caryopsis are sticking so tightly to the grain that it's just uncomfortable to eat. And And there is so responsible for this hull phenotype is a kind of glue between the pericarp and these husks. And here we knew about the gene which is responsible to switch on the biosynthesis pathway forming the glue, which is necessary to stick these to the grain. And so I thought it would be very interesting to create then a barley form which can be consumed, because barley actually is a very healthy food. It's more healthy than wheat, more healthy than maize. And everyone knows that oats, for instance, is very healthy. Barley is on a par with oats because it contains a lot of beta glucans, as is highlighted here, much more than the other cereals, and thereby it helps our digestive tract and also our blood system, for instance, to counteract the acquisition of diabetes. So, and as you have seen already for the 2 and 6 rod principle, we had a look at this Genome gene. So we needed to knock it out so that this glue between pericarp and husk is no longer formed. And as you see here in these two blocks, the upper line is always the wild type, the original sequence, and below are different mutations we obtained in a panel of mutated plants. And with the right-hand side, you always also see how big the modifications are and whether it's a deletion or an introduction of a nucleotide. And as you can see in the right corner above, this work has been published, coincidentally, by our translator, which is Sofia Gerasimova, and she was also part of my group at the time. So the result phenotypically is that in the wild type, you see that the grains are surrounded by these husks, which cannot really remove even during threshing. And our mutations or mutants are easily threshed and get rid of the husk, and therefore it can be directly used as human food. And as you can see in the right picture, this is an example again with the deletion of this one nucleotide, and you have the big phenotypic difference between hulled and non-hulled or naked barley. So the next example is about plant height, which is very important trait for crop species. As you can see here in this old painting from the 16th century, at that time, cereals were much taller than nowadays. So you see the guy there is actually as long as cereals. And of course, not only the cereals were larger, also the humans were smaller. So somehow the same level at that time. So the modern cultivars featuring reduced height are constructed due to mutations in very different genes, but most of them are related to the gibberellic acid pathway because gibberellic acid is a major driver of elongation of cells and elongation of plants. Yeah, and what the translator has already said, all the deeper sense of all that is that the shorter plants have a much higher lodging resistance, and you can use more nitrogen to fertilize to increase yield, and you have an improved relation with grain and straw. So all features of modern cultivars. And when you compare the plant height from a couple of 100 years ago with nowadays cultivars, you see the difference. So in this harvesting machine, there's a guy sitting, and he is sitting and still fits into this little cabin. And when you compare it to the plant height, what cereals these threshers, it becomes clear how short straw these cereals are. The same principle was actually used in almost all cereals and even other plants beyond cereals, and are nowadays the nutritional basis of modern society. So, and in barley, it was known that a brassinosteroid receptor contributes to the plant height. And a mutant was known, which is called the USO mutant, which features a smaller size than wild-type barley. So, and when we knock out this brassinosteroid insensitive gene, which encodes a protein which recognizes brassinosteroid as a signal, so the plant become blind against the signal. You see the wild type here, and only when I tell you that there are 3 more pods, you see that there is something, but almost nothing. So actually, these plants are not acceptable as crop plants. So the knockout of this gene has a very severe phenotype. So, and one trick to induce mutants which have still residual function of the gene. A mutation does not always mean that the gene function is entirely abolished. We can also try to introduce mutations where the effectivity of the gene is just reduced. And we tried that in selecting so-called in-frame mutations, where deletions are 3 nucleotide or 6 or a multiple of that, just eliminating one amino acids or some amino acids, which results in the retention of some function. And as a result, you see here a mutant which is smaller than these tricky guys here at the left-hand side, but maybe half the size of the wild. And on the right-hand side, you now see another example which comes closer to a useful crop, which is like 80% of the height of the wild type. And we just demonstrated that by this principle, we provide the breeder with with opportunity to regulate plant height to their needs, to vary this plant height and select also according to the breeding scheme, for instance, is it a line cultivar or the hybrid cultivar or regional requirements, so they can fine tune the plant height according to their needs. So the next topic is virus resistance. Of course, all organisms suffer from virus diseases, and so does barley. And one important group of viruses in barley is the bimoviruses, namely two basic form barley yellow mosaic virus and barley mild mosaic virus. And they are not infecting themselves, the plant. They need a intermediate host, which is Plasmodium polymyxin graminis. And this infects the plants via the roots, and then the virus can spread in the plant. And we have a lot of handicaps for the plants when they are infected. So, and some resistances are known against these bimoviruses, and one most effective relates to this left yellow structure within the translation initiation complex. This is a eukaryotic translation initiation sector, which directly interacts with messenger RNAs. So it contributes to the translation of plant messenger RNAs, but the virus, which has only a couple of own genes, recruits the whole mechanism to translate its own RNAs. But viral RNAs have not the typical cap structure of plant messenger or higher organisms messenger RNAs. It mimics that by forming in a proteinaceous structure. And the resistance is based on small modifications in this EIF4E gene. It's the interaction point with the messenger RNAs. And in some mutations, the system is still capable of processing messenger RNAs, but no longer of viral RNAs because they have small differences. And if the viral RNA cannot be processed, the virus cannot spread, and infection is actually resolved. So since this EIF4E is not a single gene encoding such a protein, it's a group of genes, we thought maybe we could knock out the most important one and the others may take over the function. And so we used our technology to knock out this EIF4E gene. And we got homozygous mutants, which co-segregated with full resistance. However, we abolished the function of the plant gene, and the plant gene as a functional gene because it needs the gene. Therefore, it was somehow expected. We were rather surprised that the plants looked pretty normal, but when it came to the harvest, So the yield was dramatically reduced in this case. So, and at that point, we switched to a more sophisticated method, which is not randomly introducing any mutations at the target site, but does only convert one nucleotide into the other at the predicted target motif. This technology is base editing because only one single nucleobase is affected. And for this, we need a derivative of Cas endonucleases, which are called nicases because they cleave only one strand of the double-stranded DNA. So it does not cut the DNA through, it just nicks the DNA at one strand. And the second feature is that it is coupled to um, a deaminase, which converts then one nucleotide into the other. And there are two types: the cytidine deaminases, which turns C to T's or G's, and adenosine deaminases, which converts A to G. So, and here you see this resistance gene, which encodes this EIF4E protein. And you see there are different single nucleotide polymorphisms, which are responsible for resistance in resistance allele. And we have two resistance allele, which are called RYM4 and RYM5. And these two genes have a couple of modifications. We still don't know which modifications are really responsible for the resistance. So we addressed several of these sites with these base editors. And now it becomes a little bit complicated, but when you follow me, maybe you may understand what the difference between the wild type is seen here in this comparison of these two blocks. The upper part is the wild type sequence, and we show the amino acid positions in the gene, then the triplets of the nucleotides, and the third line is the encoded amino acids. And this RM4 haplotype contains in position 57 instead of a TCC, a TTC, which encodes then a phenylalanine instead of serine. So, and this also it's now one example of what we were capable of creating. So it was possible to genocopy exactly the resistant homoallele in this position. So we had a plant which features exactly this conversion. But in the arms race between plants and viruses, where the plant maybe undergoes a modification and the virus is no longer to use this factor, but over time the virus adapts and is then breaking the resistance. we are also interested in other mutations and similar mutations. And so we consider also similar mutations we obtained as very valuable for future breeding to provide a biodiversity of these alleles, and these need to be tested whether they may also confer resistance.
So
So the same we did with another position in the gene, and here we created additional alleles which may also confer resistance, and all this is still in progress. So we have rendered those mutants homozygous, and they are currently subjected to infection assays, and we will find out which of these may confer resistance. So the next story is about food quality. So what I learned when the wheat durum genome was published, the reference genome, I learned that durum wheat contains comparatively lots of cadmium in the grains. And this is an interesting scientific story, that it was found out that the majority of durum wheats have a mutation which leads to accumulation of cadmium in the grains. And the reason is that this heavy metal transporter HMA3, which is shown here, is mutated. And the reason is during domestication that this mutation was successful because this heavy metal transporter also sequestered zinc. So it's not specific to cadmium, it also sequestered zinc in the vacuole. And this draws zinc be available for the plant. So in typical soils, it doesn't matter. There's sufficient zinc there, and this enzyme don't need to be specific to cadmium. And the plant does not suffer. And the plant actually does not suffer. But under agricultural conditions, it's of course good to make the zinc again available. But what this enzyme actually does, it prevents the cadmium to go into the stem and end up in the grains. And so by abolishing the function, cadmium is accumulating in the grains. And we set out to restitute the function of this gene. And this is, of course, another challenge. Before, we have only abolished gene functions by the kind of targeted mutagenesis approach. And here we had the challenge to restore a function. And yeah, Sophia. So in the right moment, we made an observation that not only one DNA repair mechanism is active in our cells, but also another one which takes advantage of small repeats on either end of the cleavage site. And I will explain to you how it works. So if you have a genomic sequence with small repeats, which are indicated here in green, ATGC, and once more, and a DNA cleavage appears to happen between these repeats. The opposite strands of these repeats are complementary to one another, and double-strand ends are typically subjected in cells of a 5' resection. So these two ends are now single-stranded, and they have a certain affinity to one another and will align, and any residual nucleotides flaps will be just deleted by this repair mechanism, so that we have a predictable modification. So every one of these repeats and everything between these two repeats will be eliminated, and this is predictable. And we now wanted to apply this interesting pathway to our case of the heavy metal transporter. And the heavy metal transporter, as I told you, is mutated. Maybe Sophia can.
Okay.
So, and the mutation I told you is actually a 7 base pair duplication in the durum wheat genome. So you see one repeat blue and the other red. And of course, we do not know what is the original one, but they are identical. And the challenge was to remove one of those to restore the function of the gene. And we've been lucky to find a suitable target motif in this in one of these repeats, and we wanted to apply the same principle. So we had what is indicated by the green background are repeats across these two uh, sequences, and we expected these to find one another by complementarity. Flaps are removed by internal enzymatic activity, and that should give rise to a perfectly restored 117 repeat allele. So, and first we checked in a protoplast system, and we were astonished how many of the mutations we obtained were exactly these 17 nucleotide deletions. And here I show you the result at the plant level. So when we mutate plants, we used to amplify these target regions and sequence them. So we had a lot of sequence reads. And the proportion of sequence reads you see here under reads. So you see that the wild type sequence is represented only by 74 reads, and the minus 17 deletion is represented by many more. And of course, this was very promising for us to get a homozygous mutation in such a case. And we had several others where still the minus 17 deletion was the majority of reads. And so in total, we have 52 primary mutants carrying this precisely restored HMA3 allele. And now we have homozygous M2 segregants identified, and phenotypic analysis of their progeny will show whether cadmium is less accumulated and as compared to the wild type.
So,
So maybe I suggest to skip the other topics, or shall I go on? Yeah, it will be maybe another 10 minutes or so. Yeah, shall I go on? Okay. So I actually wanted to give you also some advice if you decide to use this technology, how to do it best. And these target motifs are actually, please tell us your story. Thanks. So when you select the target motifs, there are online platforms available, and they are based on artificial intelligence. And actually, we do not exactly what criteria they used. They are just taught, and of course, with smart mechanism. But I will show you that those platforms are not very useful for us. We can believe in them, we can use them, but I guess at the moment, at least, we have better ways to find suitable target motifs. And I will also explain you in short why this may be the case. So what you see here is a couple of genes we have mutated, and the number of plants analyzed in the second column, and then the prediction of a certain guide RNA predicted by such a platform. And this is one of these most famous platforms. And in the first case, for instance, we analyzed 64 plants, and 35% of these transgenic plants were mutated. And the prediction of death chain is that 50% of these plants will be mutated. In the third line, you see a prediction of 58% of the plants will be mutated, but we were much more successful. We got 100%, it is shown in green. And at the opposite end, you see here in the AS, in the Aldostrian case, prediction was in green 69%, and we obtained only 5% mutation rate. And the line thereunder, even no mutation, even though it was average efficiency predicted. And based on that, we should select our target motifs. Then we ask the question, so what are the principles these platforms are using? And to understand that, and at the same time, we observed the discrepancy of the prediction and our real life results. There was a very interesting publication. And this is also a bit challenging, but maybe you will, if you try to follow, you may understand. You see here the comparison of like 7 of those prediction platforms, and a lot of genes mutated in the group who published that. And this is the Waterhouse group in Australia, very famous, very good guys, and smart. And the prediction levels are shown here in these colored boxes. And I just want to give you the extreme examples. So in these first 2 genes, we had predictions which ranged from 98% till 0.1%. So these platforms were very different in their predictions. And what they obtained was, in the last column, 100% mutation efficiency. So, and coming to the low efficiency cases here below, another 2 genes, again, predicted mostly to be maybe of average efficiency, and they didn't get any mutation. So of course, we ask our question why we should use these platforms. And the summary of these authors was that there was little consensus among the rankings by the different algorithms, and nor a statistically significant correlation between rankings and in vivo efficiencies, as I have explained. So, and we used to look at the modeled 2D structure of these guide RNAs, because we guess this is the most important issue in the specificity and the usefulness of target motifs, and independence of the sequence of the target motifs, these guide RNAs form. And here you see a very perfect example where these guide RNAs features 2 major structures. This is here a big stem loop at the left side, and 2 shorter stem loops. They are responsible for interacting with the Cas endonuclease, that they can really form a complex. And as is indicated from the 5' sign, 20 nucleotides are the specific one for the target sequence. In this region, you could not have too much double-strand DNA, more single-strand DNA, so that the single strand can really interact with the target DNA. And this is a bad guide where these major structures responsible for the interaction of Cas with Cas are partly abolished. One is absent entirely. And so I would predict this guide RNA not to be efficient. So, and finally, I would like to introduce you to our vector system, which we use for genome editing. You do not need to understand all details. The system starts with integrating these 20 specific nucleotides. So, Sophia, I forgot about you. I go back.
So, shooting was standard specific types. Starting with that, I have a very young Russian, production,
So these structures can be easily obtained using the two platforms for the formation of RNA structures. And so everyone can easily do it and check these structures individually. So back to the vector system, the first step is to introduce these 20 specific nucleotides, which are target specific. In the upper left corner, you see that in the guide RNA generic vector, these 20 nucleotides are introduced, and then we can, with this system, create 4 different guide RNA modules, and they are then assembled in one step. And this you see here in the third row, called multiple guide RNA expression unit. And for this part, we have different variants, different promoters driving such RNA genes, and also different scaffolds with certain features, which can be easily combined in one step. Likewise, for the Cas endonuclease module, we are flexible in using different promoters, different versions of Cas endonuclease, and also different terminators, forming this Cas endonuclease expression unit. And finally, we have a dummy position where any sequence of interest can be additionally be integrated, be it a GFP construct to indicate transgenesis or a repair template for genome editing or whatsoever. And all 3 functional units are then assembled into a simple vector, which can be used for direct DNA transfer, which we do for instance, protein transfection. And finally, we can transfer the complex set of expression units into a binary vector, which is required for oligonucleotide transformation, shown here below. So, and all our work was so interesting for our Federal Minister of Education and Research that she visited our lab. She even practiced a little bit and was very excited, as you hopefully are today. So, and with that, I would like to conclude. First of all, thanking all the people in my group And of course, thanking you for your patience and attention. Yes.
Thanks so much, Jochen, for the wonderful talk. Are there any questions? Sizlarga kattakon agar savolinglar bo'lsa, o'zbek tilida bersalar, biz ingliz tilida ularga tarjima qilib beramiz. Ozod?
Thank you very much. I'm Ozod Ryan, head of the laboratory of plant biotechnology and supply resources too. So I have a lot of questions, but I asked some of them. Is the VRS1 gene available in naturally 6-row bar?
So the wild barleys are all two-rowed. The 6-rowed barleys only, sorry. So the wild barleys are all two-rowed. The 6-rowed barleys appeared during domestication. And the two-rowed barleys actually have advantages for a wild-type barley because these individual grains are more easily covered by soil and rescued from be drying out. And the 6-rowed is a feature of cultivated barleys. And there was a debate because in some ancient land races or even in exceptions before land races, the 6-rowed feature occurred, but it was then shown that it was a reintroduction from cultivated barleys into wild genotypes. So this is very clear answer.
What about the VRS1 plants? They are quality decreased or not?
The grain quality per se is not decreased. There's only one problem. We cannot use these 6-rowed barleys for malting and brewing because the homogeneity of maturation along the racks is not even. So the 2-rowed barleys mature synchronously along the whole racks. In the 6-rowed barleys, we have some forerunners and some successors. So, and for the molding process, we need even material. This is the only quality feature which decreases.
Yes.
What about the other two of this VRC genes, which we know they are around 5 or 6? Did you target rest of genes?
We did not have the time yet, but it's a very interesting approach.
And my next question is the RR1 here. The RR gene, the coding dwarf.
Yeah.
What about the yield, grain yield, period of maturity? Are they reasons?
So the grains are only affected when the whole plant stature is too freaky. So in the longer examples, which have only a small reduction in plant height, the grain formation is fine. Thank you too.
If it's not the case, thanks so much again. Sizlardan uzun so'ragan holda so'z navbati berishda ozgina shoshib qo'yamiz. Buning uchun uzr so'rayman. Chunki bu workshopni tashkil qilishda o'sha ikkita institut juda ham katta hissasi. Biz bularni qo'llab-quvvatlashsiz bu yergacha yetib kela olmasdik. Men hozir so'z navbatini genetika va eksperimental biologiyasi instituti direktori Abdujalil Narimovga beraman. Marhamat.
Sizga rahmat. Ko'p odam so'rayapti. Siz kim? Qayerda ishlagan? deb haligi so'rashyapti. Mana rektor ham so'radilar mendan, hozir domlalar so'rayapti. Siz o'zingizni tanishtirib oling. Qaysi institutga kelgansiz?
Milliy universitet bitirgandan keyin genomika va bioinformatika markaziga ilmiy xodim ishga kirganman. Bu yerda 4 yil stajirovka qilib, keyin Aslında
Siz bularni guruhidasizmi?
men bularni anaqada konferensiyaga borganman va u yerda doklad qilganman. Umrimda birinchi marta ingliz tilida doklad qilganman. Bular уже o'sha sessiyani boshqarayotgandilar. Keyin nima qilib bular o'zlari nima desam ish taklif qildilar borib ana
Qayerdasiz?
hozir u yerda shu yil aprelda 7 aprelda bu yerda Germaniyada PhD'ni himoya qilib, hozir Amerikani o'sha Michigan davlat universitetida anaqa ishini boshlaganman. 4 oy bo'ldi. To'g'risi hozircha 2 kun uxlamay keldim. Уже 2da kelganman. Uxlaganim yo'qman. Umuman o'shanga gaplarim sal alg'ov-dalg'ov shuni tushlardan uxlayverayman lekin.
Qarang. Shunday xodimimiz bor ekan, bilmayotgan ekanmiz degan gap borki, ismingiz nima edi?
Farno.
Varrohat. Demak, mana hurmatli mehmonlarga tushuntirib, mana bu bugun hozir ham mehmonlar, chetdan kelgan mehmonlar, ham mana hurmatli domlalar o'tirishibdi, studentlar o'tirishibdi. Bularni bugun haligi nima deydi, bizni institutimizda tashkil etilgan loyiha doirasida tashkil etilyapti. Maqsad borib, mana hozir biz 5 ta xodimni chet elga yubordik. Ancha katta mablag' ketdi. Endi shu mablag'ni tejatilsin. Mana bularni chaqirdikda eng zamonaviy bilimlarni berish uchun. Ko'proq mana bu agrar universitetidagi studentlar, magistrlarga juda ham kerak, 3 kun davom etadi. Shuning uchun biroz bularga necha kun bo'ladimi, necha kun bo'ladi?
3 kun chorshanba kuni obedgacha.
Chorshanba kuni obedgacha.
Ha.
Hop, obed nima bo'lyapti?
Menimcha tashkillashtirganmiz.
Ha?
Kechki.
Yo'q, eng muhimida. Rahmat. Endi mana gapni tarjima qilib turasizda. Men hozir bir og'iz mana bu mehmonlarga aytmoqchiman, ham chet eldan kelgan mehmonlarga, ham mana bu o'zimizdan mehmonlarga O'zbekiston Fanlar akademiyasini rahbariyati ishlash kerak edi. Ming uzr so'rashdi. Menga topshirishdi, shu mehmonlarga minnatdorchilik bildirishdi va bularni tabriklab qo'yishdi, menga nima qildi. Men ham Fanlar akademiyasi rahbariyati nomidan, ham institut rahbariyati o'zim nomidan men bularni hammasini tabriklayman, hammasiga muvaffaqiyat tilayman. Tarjima qilib bering.
He's happy to have you here as a guest speakers, and one of the institute